Hello, everyone. Welcome to another episode of Deep Dive Knowledge Talk. Welcome to our discussion on the groundbreaking AI model, AlphaFold, and its profound impact on the field of structural biology. Today, we delve into how AlphaFold seamlessly integrates genomic data and cutting-edge techniques to predict protein structures, revolutionizing our understanding of genetic diseases and drug discovery. We will explore the transformative potential of this technology, its ethical implications, and the future collaborations it fosters between AI and experimental approaches. Join us as we unravel the exciting possibilities and challenges that lie ahead in the era of AI-powered protein structure prediction. What are the main challenges associated with the protein folding problem that AlphaFold aims to address? And how has the difficulty of this problem historically impacted the fields of biology and medicine?
The protein folding problem is a bit like trying to solve a really complicated puzzle, where each piece represents an amino acid. The bigger the protein, the more pieces there are, and the more ways those pieces can fit together. This makes the challenge much tougher. Historically, scientists have thought about the folding of proteins as a very time-consuming process. There's a famous idea that suggests if we tried to look at all the different ways a typical protein could fold, it would take longer than the entire age of the universe. So, you can imagine how difficult it is to predict the right shape a protein should take to work properly. Understanding how proteins fold is crucial, because their shapes determine how they function. If they don't fold correctly, it can lead to serious health issues like Alzheimer's, Parkinson's, and cystic fibrosis. In these diseases, misfolded proteins can cause problems not just for the individual, but can also be passed down to future generations. Moreover, knowing how to predict and understand protein folding can lead to exciting breakthroughs in designing new proteins that can help with big challenges, like creating enzymes that break down plastic waste. This means that AlphaFold's work is not just important for understanding diseases, but also for finding solutions to environmental problems.
Can you explain how AlphaFold employs advanced techniques from structural biology, bioinformatics, and machine learning to predict protein structures, and what specific innovations set it apart from previous methods?
AlphaFold uses a combination of techniques from different scientific fields to tackle the challenging task of predicting how proteins fold into their three-dimensional shapes. Imagine building a complex Lego structure where each piece represents a part of a protein. To do this successfully, you need to know not just how each piece looks, but how they fit together to create the final model. One of the main innovations of AlphaFold is its use of deep learning, which is a type of artificial intelligence that learns from large amounts of data, like a student studying from many books. It employs deep neural networks to predict two important things about proteins: the distances between pairs of amino acids, the building blocks of proteins, and the angles between the chemical bonds that connect them. This is like figuring out how far apart Lego pieces should be, and the direction they should point. What sets AlphaFold apart from older methods is its ability to predict entire protein structures all at once, rather than folding them piece by piece. Well, this is similar to assembling a complete Lego set directly from the instructions instead of building individual sections first, and then trying to combine them later. By optimizing the predictions through a method called gradient descent, AlphaFold can make small adjustments to improve accuracy, resulting in a highly precise model of the protein. Furthermore, AlphaFold was developed without relying on previously solved protein structures as template, focusing on predicting shapes from scratch. This approach has allowed it to achieve a much higher degree of accuracy than previous methods, making significant strides in solving one of biology's most complex puzzles.
What impact do the accurate predictions made by AlphaFold have on drug discovery and our understanding of genetic diseases, particularly in relation to conditions like Alzheimer's and Parkinson's?
The accurate predictions made by AlphaFold can have a significant impact on drug discovery and our understanding of genetic diseases, especially conditions like Alzheimer's and Parkinson. Imagine proteins as tiny machines in the body. If they are built wrong or malfunction, the whole system can break down, leading to diseases. When AlphaFold provides precise models of proteins, it helps scientists understand how these protein machines should look and work. This is crucial because many genetic diseases are caused by proteins that misfold. similar to trying to fit a square block into a round hole. For instance, in Alzheimer's and Parkinson, misfolded proteins can lead to the formation of harmful clumps that disrupt normal brain function. With better models from AlphaFold, researchers can identify the specific ways these proteins malfunction, which is like finding out exactly how a broken Lego piece prevents you from finishing your set. This understanding can guide the development of new drugs that target these faulty proteins, helping to restore their proper function or prevent them from misfolding in the first place. Furthermore, by accurately predicting protein structures, AlphaFold facilitates the design of new proteins or drugs that can interact with the misfolded ones. This can lead to innovative treatments, potentially altering the course of these diseases and improving the quality of life for many individuals affected by them. In summary, AlphaFold not only enhances our knowledge of protein functions, but also opens doors for developing effective therapies for serious genetic conditions.
Could you elaborate on how the integration of genomic data with AlphaFold's predictions enhances our overall understanding of protein functions and their roles in biological processes?
Integrating genomic data with AlphaFold's predictions is like using a treasure map to find hidden gems in a vast ocean. Genomic data provides detailed instructions on how proteins are built, while AlphaFold predicts their three-dimensional shapes based on those instructions. By combining these two resources, scientists can gain a much clearer understanding of how proteins function in biological processes. The genomic data essentially tells us the sequence of amino acids, which are like letters in a recipe. AlphaFold then transforms this sequence into a 3D structure, revealing how these proteins are supposed to look and work. This enhanced understanding is crucial for several reasons. First, it allows researchers to see how specific changes in the genetic code might affect protein folding and function, which is particularly important for understanding genetic diseases. For example, if a tiny change in the sequence leads to a misfolded protein associated with diseases like Alzheimer's or Parkinson's, scientists can pinpoint exactly where the problem lies. Additionally, with the accurate models that AlphaFold provides, researchers can explore how proteins interact with one another and with other molecules in the body. This is important for drug discovery, as it helps identify potential targets for new medications. By knowing the shapes of proteins, scientists can design drugs that fit them perfectly, much like a key fitting into a lock. In summary, the integration of genomic data with AlphaFold's predictions enhances our understanding of protein functions by providing clear insights into how proteins are constructed and how they operate in various biological processes. This knowledge ultimately helps in developing new treatments and understanding the underlying causes of genetic diseases.
What future developments in AI and computational biology do you foresee that could further enhance our ability to predict protein structures or even expand the applications of such predictions in various scientific fields?
Future developments in AI and computational biology hold great promise for enhancing our ability to predict protein structures and expanding the applications of these predictions across various scientific fields. One potential advancement is the continued improvement of deep learning techniques. As AI systems become more sophisticated, they could become even better at integrating vast amounts of genomic data. This would allow scientists to make more accurate predictions about how proteins fold and function, similar to using a more detailed map that highlights not just the roads, but also the landmarks along the way. Additionally, as genetic sequencing becomes cheaper and more accessible, the amount of genomic data available for training AI models will increase significantly. This wealth of data could help refine predictions by providing more examples for the AI to learn from, making it easier to identify patterns in how proteins behave. Another exciting area of development is the potential for AI to assist in protein design. By understanding the structures and functions of proteins better, scientists could design new proteins with specific functions, like creating biodegradable enzymes to tackle environmental issues such as plastic pollution. This could lead to significant advancements in sustainability and environmental science. Moreover, AI could facilitate more collaborative research across different scientific disciplines. By bringing together experts in structural biology, bioinformatics, and machine learning, Scientists can share insights and techniques that enhance our collective understanding of protein functions and their roles in biological processes. In summary, the future of AI and computational biology looks bright, with the potential for more accurate predictions, enhanced protein design capabilities, and greater collaboration across scientific fields leading to innovative solutions for both health and environmental challenges.
How do you envision the collaboration between artificial intelligence and experimental methods evolving to validate and complement the predictions made by models like AlphaFold in real-world biological research?
The collaboration between artificial intelligence, like AlphaFold, and experimental methods is set to evolve in a way that strengthens the validation and application of protein structure predictions in real-world biological research. Currently, traditional experimental techniques, such as cryo-electron microscopy and X-ray crystallography, require significant time, resources, and expertise. These methods are like trying to build a Lego model by hand, taking many trials to get each piece just right. In contrast, AI models can quickly generate predictions about protein structures based on genomic data, functioning like a well-designed template that helps guide the construction of the model. As AI predictions improve, researchers can use these models to design targeted experiments. For example, scientists may choose to focus their experimental efforts on the most promising structures predicted by AlphaFold, reducing the number of trial and error attempts needed. This way, AI acts as a powerful assistant, streamlining the research process and helping to prioritize which proteins to study further. Moreover, the integration of AI predictions with experimental methods will likely lead to a feedback loop. When experimental results validate or refine AI predictions, it enhances the accuracy of the AI models over time. Much like a student learning from their tests, this will allow for continuous improvement in both AI predictions and experimental approaches. In summary, the future collaboration between AI and experimental methods will create a more efficient and effective research environment. AI can guide experiments by providing accurate predictions, while experimental results can feed back into the AI systems to enhance their accuracy, ultimately leading to deeper insights into protein functions and their roles in biological processes.
What ethical considerations should researchers keep in mind as they utilize AI technologies like AlphaFold in biological research and potential clinical applications, especially regarding data privacy and the implications of genetic information?
As researchers utilize AI technologies like AlphaFold in biological research and potential clinical applications, several ethical considerations must be kept in mind, particularly regarding data privacy and the implications of genetic information. First, the use of genomic data raises significant concerns about privacy. Genetic information is inherently personal, revealing not only individual health risks, but also insights into family traits and ancestry. Researchers need to ensure that any genomic data used is anonymized, and that individuals have given informed consent for their data to be used. This is akin to ensuring that a diary is kept confidential before sharing its contents with others. Second, there are implications regarding the potential misuse of genetic information. As AI models become more capable of predicting not just protein structures, but also potential health outcomes based on genetic data, there is a risk that this information could be used inappropriately, such as for discrimination in employment or insurance. Researchers must be vigilant about how predictions are communicated and used, ensuring that such data is treated ethically and responsibly. Additionally, the integration of AI in biological research may lead to biased outcomes if the underlying data is not representative. If the genomic data used to train AI models comes from a limited or biased population, the predictions may not be applicable or accurate for broader groups. This is similar to trying to fit a shoe that only fits a specific foot shape onto everyone else. Researchers should strive for diversity in the data to ensure that AI predictions are equitable and beneficial for all. In summary, as AI technologies like AlphaFold continue to advance in biological research, it is essential for researchers to address ethical considerations related to data privacy, the potential misuse of genetic information, and the need for representative data. By doing so, they can harness the power of AI while maintaining the trust and protection of individuals' genetic information.
How can the scientific community ensure that advancements in AI-driven protein structure prediction, like those achieved with AlphaFold, are accessible and beneficial to researchers and institutions across different regions and levels of resource availability?
To ensure that advancements in AI-driven protein structure prediction, like those achieved with AlphaFold, are accessible and beneficial to researchers and institutions across different regions and levels of resource availability, several strategies can be implemented. First, making the technology open source or freely available can significantly increase accessibility. By sharing the algorithms and tools used in AlphaFold, researchers from all over the world can use them without needing to invest heavily in proprietary software. This democratizes access to cutting-edge technology and empowers researchers in resource-limited settings. Second, providing comprehensive training and educational resources is essential. Workshops, online courses, and tutorials can help researchers understand how to use AI tools effectively. This can be similar to offering a guided tour to help people navigate a complex museum, rather than leaving them to explore on their own. Training programs can be tailored to different skill levels, ensuring that everyone, from beginners to advanced users, can benefit. Additionally, fostering collaborations between well-resourced institutions and those with fewer resources can promote knowledge sharing and resource pooling. Partnerships can facilitate the exchange of expertise and materials, allowing researchers to leverage strengths from both sides. This collaboration can be compared to a team project where everyone brings their unique talents to achieve a common goal. And, moreover, funding opportunities specifically aimed at supporting AI-driven research in underrepresented regions can help bridge the gap in resource availability. Grants and financial support can enable researchers to engage in impactful studies, thus enriching the global scientific community. In summary, ensuring accessibility and benefits from AI advancements in protein structure prediction requires open access to technology, educational resources, collaborative efforts, and targeted funding initiatives. These steps can help create an inclusive environment where researchers from all backgrounds can contribute to and benefit from scientific progress. With feel and benefit from scientific progress.
What are some potential limitations or challenges of using AlphaFold and similar AI models in predicting protein structures that the scientific community should be aware of as they interpret results and conduct further research?
While AlphaFold and similar AI models have made significant strides in predicting protein structures, there are several potential limitations and challenges that the scientific community should be aware of as they interpret results and conduct further research. One major limitation is that AI models, including AlphaFold, rely heavily on the quality and quantity of the training data. If the genomic data used to train these models is limited or biased, the predictions may not be accurate for all types of proteins. This is akin to trying to learn a language from just a few books. The more diverse the reading material, the better the understanding. Another challenge is that while AlphaFold excels at predicting static protein structures, some proteins are not always static. They can change shape based on environmental conditions or interactions with other molecules. This dynamic nature can be difficult to capture in a model, meaning that predictions might not fully represent how proteins behave in real biological systems. Additionally, the results produced by AI models need to be validated with experimental methods. However, traditional experimental techniques, such as cryo-electron microscopy or X-ray crystallography, can be time-consuming and resource-intensive. As a result, there might be a lag between prediction and experimental confirmation, which could slow down the pace of research. Moreover, although AlphaFold predicts the overall 3D structure of proteins, it does not necessarily provide insights into the functional implications of those structures. Understanding how a protein structure relates to its function is a complex challenge that requires further investigation. Lastly, researchers should be cautious about over-relying on AI predictions without critical evaluation. It's essential for the scientific community to maintain a balanced approach that combines AI-driven insights with traditional experimental methods and biological understanding. In summary, While AlphaFold offers powerful tools for predicting protein structures, it's important to recognize the limitations related to data quality, the dynamic nature of proteins, the need for experimental validation, and the interpretation of functional implications. By being aware of these challenges, researchers can more effectively utilize AI in their biological research.
Thank you to our esteemed guest and our wonderful audience for joining us today. As we conclude our discussion, let us recap the key points. AlphaFold, an AI marvel, revolutionizes protein structure prediction by harnessing genomic data and a symphony of scientific disciplines. This breakthrough has profound implications for drug discovery and unraveling the mysteries of genetic diseases. The seamless integration of genomic data provides invaluable insights into protein functions and their roles in biological processes. With AlphaFold, we unlock the potential to diagnose and treat diseases with unprecedented precision. The future holds immense promise as AI and computational biology continue to evolve. We anticipate even more accurate predictions, the ability to design proteins with desired properties, and the fostering of cross-disciplinary collaborations. Leveraging the synergy between AI and experimental methods will accelerate validation processes and enhance research efficiency. While we embrace these advancements, ethical considerations demand our attention. Data privacy, potential misuse of genetic information, and data representativeness must be carefully navigated. To ensure equitable access and benefits, we advocate for open-source technology, accessible training resources, diverse collaborations, and targeted funding. AlphaFold's journey is not without its limitations. Reliance on training data quality, challenges in capturing protein dynamics, and the need for experimental validation should be considered when interpreting results and conducting further research. Our scientific community must thoughtfully address these limitations, paving the way for continued progress and transformative discoveries. Thank you once again for being a part of this enlightening dialogue. Together we can harness the power of AlphaFold and its transformative potential for the benefit of humanity.